Welcome. From the last video, we are learning about the methods to study global gene expression analysis. In that, we have came across transcriptomics, which is one of the strong technique for the global gene expression analysis. Transcriptomics have still some disadvantages which have created a need to develop the new method, and that it is a proteome or the proteomics. Let's see first what are the disadvantages of the transcriptomics. Transcriptome has some of the disadvantages in that the transcriptome it may not correspond to the total content of the protein. Now, when you have learned about the gene expression, what is the gene expression? In the gene expression, there is a transcription and the mRNA it is going to synthesize, and from that mRNA, there will be the synthesis of the proteins, that it is a translation. At the major point or at the majorly, when you say the gene expression means the protein synthesis. Okay, now here when there is a study of the transcriptome or when there is a transcriptomics, in that the total transcript which are present, that transcript it may not represent or it is not going to correspond to the total proteins which are present into the cell, and this is due to the firstly due to the post-transcriptional gene regulation. Now here there is the example of the oocyte. Okay, in the oocyte there is the presence of the mRNA, and that mRNA, they are some of the mRNA in the oocyte, they are the masked mRNA, and as they are the masked one, they are not getting translated. So here the mRNA from the cell, it is going to be analyzed with the help of the transcriptome, but that total mRNA, they are not going to represent the total proteins which are present into the oocyte. Second, it is the protein diversity, and that protein diversity, it is going to be due to the post-transcriptional modification, and here that post-transcriptional modification, it is the alternate splicing and the RNA editing. Here we will take the example of the alternate splicing, and that it is the large T antigen and the small t antigen of the sp14. So here it is a gene for the antigen, and this it is a single gene from where there is a synthesis of the large T antigen and the small t antigen. Okay, so here when you look at this gene, you can see that there is exon 1 and there is exon 2, and here in between, okay, here in between there is what? There is the intron, and in the intron there is the presence of the stop codon. Now here in the first case, here there is a synthesis of the primary RNA. Okay, this it is a primary RNA, and here there is a no splicing. No splicing, that is this intron which is present into this primary RNA transcript, it is not removed. Okay, so what is going to happen? This mRNA, this mature mRNA which is here, it is going for the translation and it is allowing the synthesis of the protein. And as at this point, okay, at this point there is a stop codon, there is a synthesis of the protein, and that protein it is of the smaller size, and so it is called as a small t antigen. Okay, now here in the next case, okay, here from the same gene there is a synthesis of the primary RNA transcript. So here you can say that both of the primary RNA transcript, they are the same one. But here there is a splicing, okay, there is a splicing of this intron, and exon 1 and exon 2, they are joined together, and this mature mRNA, now it will go for the translation and it is allowing the synthesis of the antigen, and that it is called as the large T antigen. So here, due to the post-transcriptional modification, that it is here, it is alternate splicing from the single gene, okay, there is synthesis of the two types of the proteins, and here the mRNA, okay, the mRNA which is going to be there, from that mRNA, with the help of the alternate splicing from the single mRNA, there is generation of the two types of the proteins after the alternate splicing. Okay, next here it is an example of the apolipoprotein B, and this it is an example of the RNA editing. Okay, there is editing, there are some of the changes into the codons after the transcription. So here it is the pre-mRNA. Okay, and in this pre-mRNA, here it is exon number 26. Okay, and in this exon number 26, CAA. Okay, there is a codon, it is a CAA codon, and it is of the 2153. The number ranging it is 2153. So here from the pre-mRNA, the mRNA, it is going to be synthesized. Okay, that is splicing has been done, and after splicing there is the mRNA. Now from the pre-mRNA, with the help of the splicing, there is a now synthesis of this mRNA. Now here this apolipoprotein, it is present into the intestine as well as it is present into the liver. But when it is present into the liver, it is of the 4563 amino acids, and when it is present into the intestine, it is of about the 2153 amino acids. That is the isoform of the apolipoprotein, it is present. One which is present into the liver, second one it is present into the intestine. So when this mRNA, it is going for the translation into the liver, at that time it is translated, and a longer apolipoprotein, it has been synthesized. Okay, and here you can see that the CAA, which is present into this pre-mRNA, it has been kept as it is, CAA. Okay, now here in the second case, when it is in the intestine, when it is in the intestine, this C, okay, this C, it is converted into the U by the site-specific deamination. So there is a deamination of this C, that is the cytosine, it has been deaminated, and when it is deaminated, it is going to convert into the U. So at this site, okay, in the exon number 26, okay, the codon CAA, now it is a UAA, which is a stop codon. So when such type of the mRNA, now it will go for the translation. Okay, it is allowing the translation, but it is going to stop at this exon number 26. So the smaller protein, which is having the amino acid 2153, it is synthesized. So it is a smaller one. And now when you look at the functions of these both of them, in the liver, it is allowing the transportation of the cholesterol and triglycerides from the liver, and in the intestine, it is allowing the absorption of the dietary lipids. So in this way, here there will be the recognition of the mRNA from the transcriptome. There will be, when you are going for the transcriptomics or when you are analyzing the transcript, you can find out the mRNA, but from that mRNA, there is there is a synthesis of the two proteins. It is not going to be analyzed with the help of the transcriptome. Okay, next here, diversity of the protein, it is generated by the post-translational modification. There are the glycosylation, phosphorylation are there, and also there are some of the post-translational modification which are allowing the cleaving of the proteins. Like here, the post-translational modification of the insulin. Here it is a pre-pro insulin. Okay, signal peptide is there. Okay, signal sequence, it is going to be cleaved. There is a synthesis of now the pro-insulin. Then the pro-insulin, it is getting cleaved. There is a cleavage, and 25 amino acids, they are cleaved from this pro-insulin. So the mature insulin, it is going to be formed. That is when there is a need of the insulin. Okay, when there is a need of the insulin, at that time only the pro-insulin, it is getting processed, and then there is the mature insulin. Okay, so here, okay, so here, though you have the total content of the protein, abundance of the proteins you have, okay, though you are observing that there is the presence of the transcript, okay, there is a presence of the proteins into the cell with the help of the transcriptome, you cannot say that what is the level of the protein activity. Okay, so the level of the protein activity, it is also has to be determined. Okay, and for this all purposes, okay, all purposes, with the finding out the localization and trafficking between the compartments, there is a need to study the proteins as it is. Here The function of the protein may also depend on its localization and the trafficking between the compartments in the regulatory manner. Now here we will take the example of this NFK beta. Now NFK beta, it is a transcription factor, and when there is a no incoming of the antigen, at that time NFK beta, it is located into the cytoplasm. Okay, and it is masked by the IK beta. Okay, so when there is no antigen localization, it is in the nucleus. When there is an incoming of the antigen, at that time IK beta, it is removed, and the NFK beta, now it is active and it is located into the nucleus. Okay, and there it is going to interact with the DNA and it is allowing the transcription. So here, okay, the protein function, its localization, and localization, its trafficking between the compartments, it has also to be analyzed. And for that purpose, there is a need to study, okay, there is a need to study the proteome or the proteomics. Now, what is the proteome and the proteomics? Proteome can be defined as the overall protein content of a cell that is characterized with regard to their localization, interactions, post-translational modifications, and turnover at a particular time. Here, the word particular time, it is very important because the gene expression pattern, it is going to change from time to time. As the environment, it is going to change, the expression pattern, it is going to change. Expression pattern, it is going to change means what? The protein content of that cell, it is also going to change. Now, so the proteome, it is going to be termed as the Homo sapiens proteome, or an organ proteome like the liver proteome, or it can be named according to the cell. And this proteome, it is not constant, it is going to differ from cell to cell, and also it is going to change over the time. Now, going towards the definition, proteome. Okay, the proteome can be defined as the overall protein content. That is, whatever may be the proteins which are present into the cell, okay, that it is going to be analyzed, and these are analyzed with regard to what? With the localization. That is, what is the location of the protein? Does it is present in the membrane, it is present into the cytosol, it is present into the nucleus or mitochondria or Golgi or in the ER, that it is going to be studied. And also the protein-protein interaction, how the proteins, they are going to interact with each other. Like how you can say that antigen-antibody interaction, okay? NFK beta interacting with the IK beta, NFK beta interacting with the DNA, that it is going to be studied. Post-translational modification like the glycosylation, then the phosphorylation, methylation, acetylation. This post-translational modification, it is also going to be studied during the proteome, and also the turnover of the proteins, it is also going to be studied. So here the proteome, it is not only going to analyze the total content of the protein of a cell, but also with that it is also going to analyze where the proteins they are located, how the proteins they are interacting to each other, what are the post-translational modifications they are occurring on the protein, and what is the turnover of the proteins. It is going to be in a proteome. And last scale study of these proteomes, it will be the proteomics. And this proteomic word, okay, this proteomic term, it is coined by the Mark Wilkins in 1996. And here, why proteomics, or what are the applications of the proteomics? Yes, I have studied the proteome, okay. I have studied its localization, I have studied interactions and all things regarding the protein. I have studied all these things, but where I have to use this, okay, it will be used for the disease diagnosis. Now, when you are studying the proteome of the cell, okay, at that time, by the comparative analysis of the two types of the cell, suppose the disease cell or any disorder is there, and on the normal one, okay, you are going to compare the proteome of these two, okay, two persons. One one it is having the disease condition, another it is a normal one. So here you can find out what is the difference between the proteins present into the two types of the cells, and you can find out the biomark. Okay, biomark, and accordingly that biomark you can you can you can do the disease diagnosis. Secondly, the level of the proteins which is present, okay, into the deceased person, that level of the protein, it is, it can be used, okay, it can be used to monitor the disease development, for the prognosis and also for the monitoring the disease development. Does the condition it is becoming a severe one, or the person it is curing, it is going to be, okay, it is going to be analyzed with the help of the proteomics. And next, and the major one, it is for the development of the drugs. Okay, so if you know the target molecule, if you know the target molecule, which target molecule, accordingly that target you can design a drug. um, okay, so in this way, these are the applications of the proteomics. Okay, so I think you may have cleared the idea about the what are the disadvantages of the transcriptomics during the global gene expression analysis. So how the proteome and the proteomics, they are going to overcome the disadvantages of the transcriptomics. So if you like my video, please subscribe my channel and also share my videos with your friends.